Spirochetes. These are helical curved bacilli detected by dark field microscopy, diagnosed by serology and silver staining. Three of them are very important. Number one is Treponema, Borrelia, and Leptospira. Leptospira is animal pea-borne pathogen, and it causes leptospirosis, well disease, and also called the jaundice. The bacterium can be isolated on Fletcher or Straught media found early in the blood and CSF, but after the second week it is in the urine, so the urine antigen test is used to diagnose leptospirosis infection. Number Treponema pallidum is a person-to-person transmission. They cause syphilis. Syphilis is a chancre at the place of the inoculation, skin and mouth rashes, CNS disorder. Diagnosis is done by the specific test, that is FTA antibodies, TPPA antigen, and are used to confirm the infection, and non-specific tests are RPR, VDRL, which monitor the infection. The last one is Borrelia. Borrelia is Burgdorferi. It's we will discuss that one in detail. Is a tick-borne infection, and they cause the lymph disease. Borrelia is epidemic. Relapsing fever is also another. The bull eye rash, flu-like symptoms, CNS and heart involvement, arthritis is also caused by this Borrelia species, which is tick-borne. There is some obligate intracellular parasites like Rickettsia, Chlamydia, Borrelia, Coxella burnetii, Francella, Ehrlichia, and Listeria. We will discuss all of these bacteria one by one. First, we will discuss the Listeria. Listeria is a catalase positive beta hemolytic and motility and bilirubin positive. Listeria is an intracellular organism, show the umbrella shaped growth in the motility agar at room temperature, and one of the opposite organism, which is sucrose negative and H2S production positive and catalase is negative, that is erythropoiesis, is a gram positive rod, cause the skin infection. It's from the fish. In motility agar at room temperature, this organism show the opposite growth of Listeria monocytogenes, and that is some brush-like shape, or you can say the opposite side of umbrella. But this organism is unable to grow at 4 degrees centigrade, erythropoiesis. So Listeria monocytogene is the major pathogen in Listeria family, and this organism, as we discuss, is motile and catalase positive, bilirubin positive, and gram positive rods, tumbling motility, listerolysine O is extracellular enzyme. 22% of neonatal death is because of this Listeria monocytogene. There is one lithium chloride, phenylethanol, maxolactam is polymyxin B, is a drug of choice for this organism. Listeria monocytogene have two more species, Listeria evonavi, which is beta hemolysis, and Listeria enochoa, which is non-hemolytic and not a human pathogen. Enochoa is a pathogen of in HIV one patient. Mostly it is seen in systemic infection in the humans. Another organism in this group is Borrelia. As we discussed, the Borrelia is Burgdorferi is Sensosteco is a US and European country, and it causes the lymph disease. It's an arthropod burn. Borrelia recurrentis is another species. Borrelia rhelodotoni is another species. All of them has caused lymph diseases, relapsing fever, loss burn disease due to the poor hygiene, crowding. Organisms are maintained by the passage between the lice and the human. In Borrelia infection and the Lyme disease, the high fever, muscular pain, headache, weakness episode usually ends suddenly after 3 to 7 days because the human response is mounted. Spirochetes disappear. Usually Spirochetes can be detected in the gems, right silver stain, and after several weeks later, the reoccurrence of disease with less severe courses occur. This relapses are because of the variation of the surface antigen of the organism, resulting in the temporary avoidance of immune system. In lymph disease, the IgM antibodies can be developed after 2 weeks of the infection, while IgG may not be detected after 3 to 6 months. And both IgM and IgG antibodies may be persist for up to 20 years after the infection. As you know, Treponema pallidum causes the venereal syphilis, and treponema is obligate human parasite, STD, active primary for syphilis, secondary lesions, and corkscrew motility. The gold standard test is a rabbit infectivity test for Treponema pallidum detection. Pasteurella maltosida is a common upper respiratory tract of the mammalians. It's a capsulated bacterium, usually cause the infection by animal biting, especially in the cats, the localized cellulitis and lymphadenitis, pulmonary disease, meningitis. The organism is susceptible to penicillin 10 microgram disc, which zone of induration is more than or equal to 15 mm at 0.5 McFarland turbidity standard. The exudate from the bite site, chocolate and SB, is the drug of the choice of a medium, a small gray smooth mucoid non-hemolytic non-motile gram-negative polymorphic cocobacilli, and the organism is catalase, oxidase, nitrate, indole, and alkaline phosphatase positive. Bartonella is a blood and tissue specimen in organism is a motile. It is also curved, aerobic, gram-negative bacilli required the enriched medium, and calciphore-like colonies, irregular, rise, wide, dry, rough colonies. The organism is oxidase and urease negative, do not produce acid from the carbohydrate, but the organism have ability to tendency to pit the agar. Three main species. Number one is Bartonella henseli, endemic infection of a domestic cat. The fleas are Catenocephaliates felis is causing the cat scratch disease. The optimum temperature for the growth is 35 to 37 degrees centigrade. Bartonella contana is transmitted by the pediculus human, worldwide distributed, also called a trench fever or 5 days fever, 35 to 37 degrees centigrade. The Bartonella bacilliformis is localized to the Indis sandfly vector and penetrate the RBCs. RBCs are fragile, and that is clear by the reticular endothelial system. So the organism is able to cause the severe anemia. Optimum growth temperature is different from the Contana and Hensela is 25 to 30 degrees centigrade, while Contana and Henseli is 35 to 37 degrees centigrade. Another species, Aphifia, is similar like the Bartonella species, but oxidase and urease positive, and also produce acid from xylose. Bartonella can be identified by the gas liquid chromatographic of their fatty acids. Pasteurella maltosida is a capsulated organism. One thing is I want to add now is TSI. On the TSI, triple sugar iron, the organism give acid by acid reaction with no gas. Francella tularensis is an extremely infectious agent, and 10 reciprocate bacteria is required for the GI infection, and 10 bacterium is more than enough for subcutaneously caused infections, and 25 bacterium is enough for respiratory route infection. Francella tularensis is transmitted through the wild rabbit, ticks, mosquitoes. Francella tularensis have the ability to penetrate the normal skin. Small polymorphic gram-negative rods, weak catalase, positive oxidase negative, 35 degrees centigrade, under 5% carbon dioxide is required. It is a alpha hemolytic, 2 to 5 days. The growth appears, which is white to greenish colonies. The organism required some specialized medium like cysteine hot agar supplemented with the 9% chocolatized sheep blood and BCYE, and another medium is a tire martin agar. Specimen are mostly scraping of the ulcer or lymph node biopsy. The clinical presentation for this organism is fever, chill, headache, generalized pain, lymphadenopathy with the cutaneous ulcers. There is ulcer glandular and glandular. Glandular is mostly in lymphadenopathy only, but ulcer glandular is just a tick bite, especially in the USA. 3-10 days, papula eventually ulcerative after 3 to 10 days. IgG up to 10 days, it is detected after 10 days, and IgM may be longer period up to many years, persistent in the blood after the infection. Direct fluorescent antibody and immunohistochemical staining using the monoclonal antibody are useful to detect the fluorescilla in the tissue. And we can also perform the PCR. In the PCR, specimen must be collected in guanidine isothiocyanate buffer, which preserves the DNA for the weeks, for several weeks. Rickettsia has 4 species, major 4 species. The major species Rickettsia rickettsi, most common in USA. It causes the Rocky Mountain spotted fever. The reservoir is tick. These ticks are Derma center Indorsoni and Derma center variabilis. Mostly infection occur in the April and October. Chill, headache, abdominal pain, vomiting, malaise, rash, fever. The rash usually started from the extremity, like palm, soil, and then trunk. In Rickettsia family, there is another subspecies that is Orentia tustogomashi. They cause the scrub typhus, and mostly found in the Asia and Australia. The mite act as a reservoir and vector. In this case, this infection, the rashes start from the trunk and spread to the extremities. So another disease is typhus, which is caused by the Rickettsia prowazaki, transmitted to human through the human body loss. Pediculus human. Lost burn typhus. Initial symptoms are similar like Rocky Mountain fever, and reoccurrence of the disease is called Brill-Zinsser disease. In Rickettsia family, there is another species that is Rickettsia typhi, that cause the endemic or murine typhus, in which the rash is restricted to the chest and abdomen. The vector is rat flea, that is Xenophilia chupus. A clinical representation is similar to the Rickettsia rickettsii or Rocky Mountain fever. The diagnosis is done by the Will-Flex test, in which the employ the protein antigen used for the many years in serological testing for presumptive diagnosis of rickettsia infection. Another diagnosis method is a punch biopsy specimen from the skin lesion, culture, immunochemistry, molecular analysis, and also can be isolated on the tissue culture, embryonic egg, virulines, and L929 cell lines, and MRC5. Another organism is new Rickettsia senso. It caused the Santoso fever, occur in the Japan by ingestion of a raw fish, and it is also called mono-casing disease. Glandular fever, lymph node enlargement, and this is because of the ingestion of a raw fish that contain the new Rickettsia, mostly occur in the Japan. The clinical representation is similar like the infection mononucleases. Chlamydia. Chlamydia is obligate intracellular bacteria. All the chlamydia exist in two forms: elementary body and reticulate body. There are three major species that cause the infection and are medically important. Number one is trichomitis, chlamydia trichomitis, and the other one is chlamydia pneumonia, chlamydia pesticii. Both of these, chlamydia pneumonia and chlamydia pesticii, cause the pneumonia-like infection, and that's why it is called chlamydiophilia. Chlamydia pneumonia is transmitted between the respiratory secretion, common cause of the community acquired in pneumonia, which lead to the asthma, esterocolosis, and strokes, Alzheimer's disease, multiple sclerosis, and chlamydia pesticii is a primary animal pathogen. A human infect in some rare cases from the domestic birds. It causes pneumonia, can lead to the fetal systemic infection. In chlamydia trachomitis, which is mostly known for the STD, and sometimes it is known for the preventable blindness. In this trachomitis, there is serotypes like KD, L1, L2, and L3. L1 and L2 and L3 are from the Africa strain, and it cause the STD, and also the D cause the STD infections disease, and sometimes it cause the preventable blindness. In the female, STD lead to the infertility in the serotype D and K. All the chlamydia exist in two forms: EB and RB, elementary bodies and reticulate body. Elementary bodies is about 0.3 micron meter and extracellular spore-like infective stage which enter into the susceptible host, located in the cytoplasm inclusions, undergo some structural and metabolic conversion and convert into the RB. RB is almost 3 times the size of EB. So metabolically active form, again the binary fission take place and go some maturation. At some point it's reorganized and back into the EB. This cycle takes 36 to 72 hours, and EB again infect the other adjacent cells, and the infection goes on. Laboratory diagnosis is a NAT, is a gold standard test, nucleic acid amplification test. Cellular cell culture cell line can also be used to isolate the organism. 2-sucrose phosphate swab is used to isolate the organism, and we can use different cell lines like HeLa, Macaque, HEPA2, HL. So Ehrlichia is one organism that is a zoonotic agent, and reservoir in this infection is tick bite, and Ehrlichia chaffinensis is a human monocytic chelerosis. In this infection, the lymph node become enlarged. Chlamydia-like organism produce the intracellular inclusions like elementary bodies and reticulate bodies, cell lysis, and infect the adjacent cell. The diagnosis is blood in EDTA, Giemsa, right stain can be detect the intracellular organisms. Moral it's inclusions, and from the CSF you can use the cell lines DH82, THP1, and virusal, ELISA, and PCR. And in the Giemsa stain, the intracellular organism morel like inclusion bodies can be detected. Brucella is a zoonotic agent acquired by the human by ingestion or skin or mucosal contact, or mostly from the ingestion of the dairy milk products. The brucella cause remittent fever, undulant fever, which is also called Bengas disease, may last for the years. The basic cause is brucella. Brucella have 4 species. Brucella militensis is agent of goats and sheep. and Brucella abortus is from the cattle, and militensis is from the Latin America and Asia, while abortus is worldwide distributed and major pathogen of cattle. And Brucella suis is from the swine and mostly found in the Asia and Latin America, US. The organ of reticular endothelial system are frequently infected, and from non-kisting granulomas, difficult to distinguish between the sarcoidosis. Brucella is aerobic, non-spore forming, non-encapsulated, non-motile, intracellular gram-negative cocobacilli. Blood, bone marrow, biopsy are the major specimen. Commercially available blood culture system with the Castaneda medium is a choice of medium, 35 degrees centigrade. Blind terminal subculture is recommended. Minimum 21 days is required for the growth. The rapid urease reaction within 1 hour, and the organism is also the drug of choice are ciprofloxacin, tetracycline, and streptomycin. This organism Brucella is classified as a type 3 biohazard. Serological testing is recommended in all cases of suspected infection. The organism is also positive for catalase oxidase urease, which is rapid, and nitrate. IgM appear in the first week, while IgG found by 2 to 3 weeks. After 2 to 3 weeks, identification of bacterial colony can be done by basic foxine, thionine, and H2S. Brucella militensis and abortus both are resistant to basic foxine, but abortus is sensitive to thionine and also have ability to produce the H2S in 2 to 5 days. Similar like Brucella suis and Brucella canis, both are sensitive to basic foxine and both are resistant to thionine, but only suis have ability to produce H2S in 2 to 5 days. Q fever divided by binary efficient can survive many years under the harsh environmental condition. It's from animal like cat, dog, rabbits are main reservoir. Tissue culture like Vero and shell walls, specific IgM and PCR is also helpful in diagnosis, animal inoculation, and embryonic egg yolk inoculation. Coxella burnetii is transmitted through aerosol, contaminated milk from the cow or goat, atypical pneumonia.